introductions of livestock animal cloning. What if livestock animal cloning? Clone can mean different things to different people. In molecular biology, it refers to the process of making progenitor cells to obtain a population of genetically identical cells, whereas in animal biology, cloning refers to the production of genetic copies of individual animals using nuclear transfer. In the farming sector, the practice of making genetically identical copy of animal is known as the livestock animal cloning. It includes creating children with the same genetic makeup as the original animal being cloned utilizing cutting-edge reproductive methods.
Next, let's go to the history of the method. Here what you need to know. The system of cloning livestock animals has a long history, and reproductive biotechnology has made amazing progress in that time. Here is a synopsis of the major turning point in livestock animal cloning history. The first history is cloning sheep. In 1996, Dolly the sheep, a fin-dorsal goat, made history in 1996 as the first mammal cloned from other somatic cells through the somatic cell nuclear transfer (SEN1A) technology. It will bring new possibility for cloning using the different cell because it's produced from the mammal plant cell of other female. The second history is clone piglets. In 2001, the first clone piglet was born. Using the process of the somatic cell nuclear transfer (SEN2), researchers from the University of Missouri and biotechnology firm Advanced Cell Technology (ACT) successfully cloned several piglets. The results show that pig cloning is possible and likely to be used in various fields such as agriculture and biomedicine.
The first conclaves were born. In the same way, the first conclaves were born in 2001. Somatic cell nuclear transfer (SCNT) was used by Texas A&M University research to clone multiple claves. These conclaves both introduced an achievement in the cloning of large animals and improved our understanding of the cloning process. Cloning for breeding elite animals. In 2015, cloning techniques were more frequently used to generate exceptional animals with desirable features. This use of cloning attempts to create genetically exact duplicates of valuable animals that had remarkable traits like superior milk output or illness resistance. The main goal was to conserve and spread particular genetic features that were regarded as beneficial in the livestock sector. With that, thank you.
Application. The cloning of an animal has been used in a number of different applications. Animals have been cloned to have gene mutation that help scientists study disease that develop in the animal. Livestock like cow and pig have been cloned to produce more meat or meat. Clone can even resurrect a beloved pig that has died. An example of a clone ruminant, Mira, was cloned in 1998. Mira and her sister were designed to express a substance called recombinant hormone and thrombin in their milk. This is a protein consistent in their milk that prevent blood clotting in our system. Kaga and her twin, Notter, will produce using a somatic cell nuclear transfer technique in which genetic information of an unfertilized its donor is replaced with DNA from a adult body cell. This cow were cloned in 1998 and duplicated several times. Made in Japan, the cow paved the way for other clone engineers to produce better meat and milk.
Advantage of livestock animal cloning. Firstly, it will boost reproduction in order to more efficiently produce safe and wholesome food. Producer can use cloning to speed up the production of their most productive livestock. Secondly, cloning could benefit consumers. Cloning can benefit consumers since milk and milk will be more nutritious and safe. Third, breeds the healthiest animal, the least amount of antibiotics, growth hormone, and other drugs are used since cloning only reproduces animals that are well. And lastly, save endangered species. Endangered species can be preserved through cloning. For instance, panda cells are being stored in China in case the species number are threatened with extinction.
Next, we turn to disadvantage. Firstly, low success rate. Cloning farm animals is a costly operation with low success rate. 1 to 5% to attempt to create viable clones are successful. Due to its poor efficacy and creating a single viable clone is expensive and taking time. Second, health issue. Cloned animals frequently exhibit a variety of imperfections and health problems. Compared to normally produced animals, they are more prone to growing defects, organ malfunction, and immune system problems. These health problems can cause cloned animals to die off more frequently and have a lower overall welfare. Lastly, cost and access. Cloning farm animals cost a lot of money and requires specific machinery and trained staff. Its high price prevents it from being widely used in the livestock business and prevents it from being affordable for many farmers.
This is discussion of case study. The study aimed to improve the genetic engineering of pig by combining somatic cell nucleus transfer with CRISPR microinjection. The researcher investigate whether microinjection of clone pig zygote following intensive manipulation of oxide would compromise their developmental competence. The researcher used fibroblasts from an Osabo pig fetus and performed SEN1A by injecting the fibroblast nucleus into an inoculated egg. They then microinjected the concentrated zygote with 2-guard RNA, targeting a specific gene called GRB10. The sgRNA were pre-complex with just 9 proteins for efficient gene targeting. The microinjection of the zygote did not significantly affect early curvature of overall development rate compared to uninjected control. The micro-injected embryos were transferred to surrogate animal, resulting in a successful pregnancy and the recovery of sick fetuses. Analysis of the fetuses confirmed that all of them carry the desired genetic modification in the GRB10 gene. No evidence of mosaicism was observed, indicating the high efficiency of the CRISPR ribonucleoprotein complete microinjection. The researcher concluded that the direct cytoplasmic injection of CRISPR ribonucleoprotein complex into zygote produced by SEN1A enable rapid and efficient gene targeting in specialty pig breed such as Osabo pigs. This combined technique eliminates the need for donor embryos from specialty herds, cell culture screening, and the occurrence of mosaicism in the resulting offspring. Overall, the approach described in the study offers a cost-effective and resource-saving method for generating pigs with specific biallelic gene edits using SEN1A and CRISPR technology.
Method used in the case study. This first one is somatic cell isolation. Collect somatic cells from donor pigs and separate them for genetics modifications. Okay, the next, genetics modification. Use CRISPR plus Cas9 technology to make desired genetics change in the isolated somatic cells. Prepare specific guides RNA for the target genes and Cas9 nucleus. Combine them to create a symmetric ctRNA and deliver it to the somatic cells. Reconstructed embryo creation. Obtain inulate or oocytes from female pigs. Fuse the modified somatic cell with inulate or oocyte using somatic cell nuclear transfer, resulting in reconstructed embryos. Embryo culture and development. place the reconstructed embryo in a sustainable growth medium to support their development, monitor their growth and development closely. Okay, the next is CRISPR plus CAS9 microinjection. Inject CRISPR plus CAS9 regions directly into the reconstructed embryo using microinjection. This process creates double-strand breaks in the DNA, inducing targets gene editing. And lastly, evolution and screening. Assess the efficiency of genome editing by examining the clones, speaks embryos for successful gene modification. Use molecular technique like PCR or sequencing to confirm the desired genetic change. Screen the clone speak embryos to identify individuals with the desired genomics modifications.
Now, I'm going to present about how livestock animal cloning helps to improve something related to natural resource. First, the disease resistance. By genetically engineering livestock animals such as pigs to be more resistant to disease, the reliance on antibiotic and other medical treatment can be reduced. This helps preserve the efficacy of antibiotics in both human medicine and veterinary medicine, ensuring the availability for critical needs. Next are the environmental impact. Genetic engineering can also contribute to reducing the environmental impact of livestock farming. For example, by modifying genes related to nitrogen or phosphorus utilization, animals can produce less waste containing these nutrients, thereby reducing the pollution of soil and water resources. Similarly, genetic modification can be targeted to reduce methane emission, which is a significant contributor to greenhouse gas emission from livestock. Last but not least are the conservation efforts. Genetic engineering can aid conservation efforts by preserving endangered or threatened species. In some cases, genetic material from endangered animals can be preserved and used for SEN1A to produce cloned individuals that can contribute to the preservation of genetic diversity and prevent the extinction of species. Thank you.